So if you think of cancer metastasis as like a high-stakes heist, I mean, we've pretty much always assumed the tumor acts alone, right?
Right, yeah, like it's just brute force breaking and entering.
Exactly, smashing through the windows. But we are doing a deep dive today into a fascinating new molecular biology paper from Cell Death and Disease, and it basically exposes the inside man.
It really does. It's a total paradigm shift.
It is, because we're looking at the exact microscopic mechanisms of how colorectal cancer spreads to the liver. And the completely unsettling reality is that the tumor, it doesn't just invade our blood vessels, it actively recruits them.
Yeah, it turns our own biology into accomplices. And that completely flips the traditional model of metastasis. We are, moving away from looking at the primary tumor as the sole suspect.
Right, we have to analyze the scene of the crime to see who actually unlocked the door.
Exactly. Because to give you some context, colorectal cancer is the second most common cause of cancer-related mortality globally. But, it's rarely the primary tumor in the colon that's actually fatal.
Yeah, the danger really starts when it moves.
Right. The mortality just spikes dramatically when patients develop colorectal liver metastasis, or CRLM.
And to get to the liver, those cancer cells have to pull off this process called vascular invasion, or VASCI.
VASCI.
Yeah, VASCI. They basically have to breach the local blood vessels, survive the insane physical sheer stress of the bloodstream, and then anchor into the liver tissue. We've known about this for a long time, but this specific source text we're analyzing today, it isolates the exact cellular negotiation that makes that break-in possible.
And they established the stakes right away. The researchers didn't just look at a few cells in a petri dish. They pulled massive data sets.
Real human data.
Yeah, exactly. They analyzed 229 colorectal cancer patients from the TCGA database.
That's the Cancer Genome Atlas for those listening.
Right, the TCGA. And then they validated that data with a 40-patient cohort from their own hospital. So we're talking high-level clinical evidence here.
Which is crucial.
Very. And the clinical data confirmed that patients with a VASCI positive status, meaning they have clear pathological evidence of tumor cells invading their blood vessels, those patients had significantly lower overall survival and disease-specific survival.
It's basically the definitive red flag for a lethal prognosis. But honestly, what I find just fascinating here is the methodology. Like how they figured out how this invasion happened.
The single cell stuff.
Yeah, because traditional sequencing, it just sort of grinds up a tumor biopsy and gives you this average genetic reading of the whole mass.
Right, it's a bulk average.
Right. But if you have like millions of cells tangled together, cancer cells, immune cells, blood vessel cells, how do you actually isolate the specific culprit that's helping the cancer?
Well, you have to move to single-cell resolution.
Oh.
So they use a technique called single-cell RNA sequencing, or cRNA-seq.
Okay, I have a visual analogy for this because I love this concept.
Oh, let's hear it.
So traditional sequencing is like taking a strawberry, blueberry, and banana smoothie, tasting it, and saying, okay, it tastes like fruit. But scRNA-seq, it's like magically taking that blended smoothie and unblending it. You separate it back into individual, completely intact strawberries, blueberries, and bananas, so you can see exactly what each piece of fruit or each cell is doing on its own.
That's actually a perfect way to explain it. Because functionally, you take the tissue sample and use enzymes to break it apart into a suspension of individual cells.
Literally unblend the tumor.
Exactly. And then, using microfluidics, you flow those cells through this tiny microscopic channel. Each individual cell gets trapped inside a tiny droplet of oil, basically a lipid droplet, alongside a microscopic bead.
And that bead has like chemical barcodes on it, right?
Yes, unique DNA barcodes. So when the cell bursts open inside its little droplet, all of its RNA, its genetic messages get tagged with that specific barcode.
Wow. So you do that for tens of thousands of cells all at once, sequence them all together, and then a computer uses those barcodes to reconstruct exactly which RNA message came from which specific cell.
Precisely. And by doing this on 8 patient liver metastasis samples, 4 that were VASCI positive and 4 that were VASCI negative, they analyzed over 95,000 high-quality individual cells.
That's a massive amount of data.
It's huge. And they mapped out 7 major cell lineages in that tumor neighborhood, T cells, B cells, fibroblasts, epithelial cells, and endothelial cells.
Okay, wait. Here is where my whole understanding of vascular biology got totally turned upside down by this paper.
The endothelial cells.
Yes. Because we typically think of endothelial cells, which are the cells that form the actual structural lining of our blood vessels. We think of them as passive infrastructure.
Right, they're just the pipes.
Exactly, just the plumbing. But the data here showed they were the most dynamic players in the entire tumor microenvironment.
It is totally counterintuitive. To prove this, the researchers ran all that single-cell RNA data through a machine learning algorithm called Augur, A-U-G-U-R.
Like a fortune teller.
Right. And Augur doesn't just look at what a cell is. It calculates how much a cell's internal programming changes in response to a specific condition, in this case, the presence of vascular invasion.
And what did Augur find?
It revealed that the endothelial cells, the pipes, were the most heavily transcriptionally reprogrammed cells in the entire tissue.
So they aren't just sitting there while the tumor pushes past them. They are actively rewriting their own genetic software in response to the cancer.
Yeah, and the researchers also used another algorithm, a network analysis tool called CellChat. It scans all the RNA data for known ligand receptor pairs to map out, essentially, who is talking to who.
Like tapping the phone lines.
Exactly like that. And CellChat showed that these reprogrammed endothelial cells were acting as major communication hubs. They were sending and receiving a massive volume of chemical signals directly to and from the malignant cancer cells.
Okay, so let's do a quick recap of what we just covered, because that is a lot of dense biology.
Good idea.
So, bullet point 1. Colorectal liver metastasis is deadly, and it relies on vascular invasion. Bullet point 2. By using single-cell sequencing, or smoothie, unblended researchers looked at 95,000 cells and found that the blood vessel cells are the most altered cells in the whole area. And bullet point 3. These blood vessel cells are actively talking to the tumor. That's the perfect summary. So my question to you, the listener, as we move into this next part is this. If you were a cancer cell, how would you go about convincing the local plumbing to just open up and let you slip into the bloodstream?
Well, it turns out the cancer doesn't force its way in. It needs the blood vessels to physically change their own architecture.
Right, which brings us to the actual rogue architects.
No.
Because the researchers didn't just stop at identifying endothelial cells. They subclustered those cells into 10 distinct subsets to find the specific inside man.
And one subset was heavily enriched in the vascular-positive samples. They named it IGFBP5 TIPEX.
Okay, let's unpack that name, starting with the TIPEC part. What is a TIP endothelial cell?
So angiogenesis, which is the sprouting of new blood vessels, it's a highly organized process. Vessels don't just appear out of nowhere. They grow, kind of like roots from a tree.
Right.
And at the very leading edge of a new sprout is a specialized cell called a tip endothelial cell. Its whole job is to probe the environment, break down the tissue in front of it, and pull the trailing stalk cells behind it to form a new tube.
So it's basically the vanguard cell. It's the point man.
Exactly. But in these tumors, these vanguard tip cells had a massive upregulation of a specific gene, IGFBP5. That stands for insulin-like growth factor binding protein 5.
That is a mouthful. Let's just stick to IGFBP5.
Yeah, good call. It's a secreted protein, which means it's a messenger molecule. These vanguard cells are just pumping this molecule out into the surrounding tumor area.
And what does pumping out all that messenger protein actually do to the blood vessel?
It induces a phenomenon called endothelial to mesenchymal transition, or END-MT.
END-MT. Okay, and from what I read, this is basically a severe cellular identity crisis.
Right. Yeah, it really is. Because healthy blood vessels, they need to be watertight. To keep that barrier strong, endothelial cells are glued together by tight junction proteins.
Specifically, a protein called VE-cadherin. Right?
Yes.
I picture VE-cadherin like a heavy-duty molecular zipper. It locks the cells tightly together so nothing leaks out.
That's a great visual. But during this END-MT process, the cells stop producing VE-cadherin.
The zipper breaks.
The zipper totally breaks. And at the exact same time, the cells upregulate mesenchymal markers, specifically proteins called vimentin and ZEB1.
And what do vimentin and ZEB1 do?
They fundamentally restructure the cell's internal skeleton. They make the cell loose, highly migratory, and spindle-shaped, so the vessel completely loses its structural integrity.
Okay, I have another analogy. It's like having a corrupt contractor.
Oh, I like where this is going.
You hire them to build a strong bridge, but instead they take out all the steel bolts, the VE-cadherin, and replace them with wet noodles, which would be the vimentin. They deliberately build a shoddy, leaky bridge.
That is exactly what's happening. But of course, the researchers had to prove this wasn't just a coincidence. They had to prove this mechanism in the lab.
Right, they had to recreate it.
So they took HUVECs, human umbilical vein endothelial cells, which are kind of the gold standard for vascular research, and they used a lentivirus to force them to overexpress that IGF-BP5 protein.
They essentially built their own synthetic corrupt contractors.
Yes, and the functional tests they ran were striking. They did a matrigel tube formation assay.
What does that look like?
Normally, if you put healthy blood vessel cells on this specific gel, they organize themselves into a really neat, highly structured capillary network, like a perfect grid of pipes.
But with the engineered corrupt cells.
Total chaos. They formed abnormally branched, physically messy tubes.
And they also did a permeability assay, right? To test if the bridge was actually leaky.
Yeah, they pass a fluorescent tracer molecule through a layer of these cells. Healthy cells keep the tracer contained, but the engineered corrupt cells, they couldn't hold the barrier at all. The fluorescent tracer leaked right through.
A completely porous bridge.
And the craziest part is what happened when they put these leaky, rogue endothelial cells in the same dish as actual human colorectal cancer cells.
The cancer cells went crazy, right?
They radically shifted their behavior, their proliferation rate spiked, and their ability to migrate across a permeable membrane just increased dramatically. The defective bridge was actively accelerating the cancer's movement.
Okay, that is wild. Time for our second mini recap.
Let's do it.
Bullet point 1. Researchers zeroed in on a specific vanguard blood vessel cell called a TIPEC. Bullet point 2. These cells pump out a messenger called IGF-BP5, which causes the zipper between the cells to break, making the blood vessel leaky and messy. And bullet point 3. This leakiness directly makes the cancer cells more aggressive.
Nailed it.
So my next question for you, the listener, is this. Now that we know the blood vessels are intentionally making themselves leaky, what do you think the tumor cells do next with this open door?
Well, there's a missing biological link we haven't talked about yet. We know the blood vessel is secreting IGF-BP5, and we know it's making the vessels leaky. But how exactly does that signal jump over to the cancer cell?
Right, because there has to be a physical receiver on the cancer cell's surface to hear the message.
Exactly. And the researchers found it. They identified the receptor protein on the tumor cells. It's called ITGB3, or integrin beta-3.
Okay, wait, I want to push back on that for a second.
Sure.
Because integrins, they're usually involved in normal cell adhesion, right? Like binding to the extracellular matrix. Why would a malignant cancer cell from the colon be expressing high levels of an ITGB3 receptor in the first place? That doesn't seem normal.
It's not normal. It's a hallmark of aggressive malignancy. Normal cells in the colon have very tightly regulated integrin expression just to keep them anchored where they belong.
Stay put.
Right. But as a tumor mutates and becomes metastatic, it frequently hijacks embryonic genes it isn't supposed to be using anymore. Upregulating ITGB3 gives the cancer cell a brand new tool to interact with abnormal environments, like that messy, leaky matrix around our corrupt blood vessel.
So the cancer cell has mutated to present this ITGB3 lock, and the blood vessel is pumping out the IGF-BP5 key.
Exactly. The secret handshake.
But how did they definitively prove that this specific key turns that specific lock? Like, how do you prove that in a lab?
They started in silico, using molecular docking software. They took the known 3D crystal structures of both proteins and used thermodynamic algorithms to calculate the physical forces between them.
So the software basically asks, energetically, do these two molecules actually want to snap together?
Yes. And it returned a docking confidence score of 0.9573 out of 1.0.
Wow. That is a near perfect fit.
It's a massive thermodynamic affinity, but as you know, a digital simulation isn't physiological proof.
Right, you need to prove it in the real world.
So they moved to physical models using a technique called a proximity ligation assay, or PLA. And this is just a brilliant piece of science.
Explain how PLA works, because the mechanism is super clever.
So you use two primary antibodies, one that binds to the IGF-BP5 key and one that binds to the ITGB3 lock. And attached to each of these antibodies is a short, single strand of DNA.
And the crucial part here is the distance, right?
Yes. Those two DNA strands will only interact if the two target proteins are physically within 40 nanometers of each other.
Which is incredibly close.
Right. If the key is in the lock, those DNA strands are close enough to bind together, forming a continuous circle of DNA. Then you add an enzyme called a polymerase. It rolls around that DNA circle, amplifying it into this massive, tangled ball of DNA.
And then you tag that ball of DNA with a fluorescent probe.
Exactly. So when they looked under a confocal microscope, the tissue literally lit up with red fluorescent dots, definitively proving these two proteins were physically touching on the cell surface.
And they back that up with co-immunoprecipitation too, right? Where they use an antibody to pull one protein out of a cellular soup and show that the other protein got dragged out along with it.
Yep, co-IP confirmed it. And then they brought this all the way back to actual human patient tissue using spatial transcriptomics, specifically an algorithm called iStar.
Oh, I was reading about iStar. It is a huge leap forward from just looking at a standard biopsy slide.
It's remarkable. It uses machine learning to superimpose high-throughput RNA sequencing data directly onto super-resolution histological images.
Meaning you can see the physical architecture of the tissue, like where the cells are, while simultaneously reading the genetic expression of the cells in that exact physical spot.
Yeah, it's like Google Maps for the tumor, but showing you what everyone is saying in every house.
Great analogy. And when they applied iStar to the human liver metastasis samples, they saw the spatial overlap perfectly. The exact geographic spots in the tissue that were expressing the IGFBP5 key were physically overlapping with the spots expressing the ITGB3 lock. Right at the leading edge of the vascular invasion. They literally caught the handshake on camera in human tissue.
They did, but having the key and the lock is only half the battle. What is the actual downstream mechanism? Like when the key turns the lock, what happens inside the cancer cell to make it so aggressive?
It activates the PI3KKT signaling pathway.
Right.
And this isn't just like a simple generic switch. It's a very specific biochemical cascade. PI3K is a kinase enzyme. So when the ITGB3 lock is triggered on the outside of the cell, PI3K gets recruited to the inside of the cell membrane.
And what does it do there?
Phosphorylates a lipid molecule, basically converting a molecule called PIP2 into PIP3.
And that PIP3 acts as a docking station for another crucial kinase called AKT. Once AKT docks there and gets phosphorylated, the cascade really takes off.
It's basically a cellular relay race. You're passing a phosphate baton down the line. And to use another analogy, this pathway is like slamming down on the cellular gas pedal.
Yeah, it revs everything up.
Because activating AKT regulates proteins that directly alter the actin cytoskeleton of the cancer cell. The cell physically changes its internal scaffolding, which generates the mechanical force it needs to push through tissue, migrate toward that leaky blood vessel, and actually survive entering the bloodstream.
But again, to prove this engine was actually driving metastasis, they couldn't just rely on cells in a dish. They had to test it physiologically.
Right, the ultimate test.
So first, they use patient-derived organoids, or PDOs.
These are like 3D mini tumors grown in the lab, right?
Exactly. They take fresh tumor tissue surgically removed from colorectal cancer patients and culture it in a 3D matrix. It grows into these miniature functional tumors that keep the complex architecture of the original patient.
And when they put those human organoids together with the rogue IGFBP5 producing blood vessel cells.
The organoids exhibited highly aggressive, invasive growth patterns, but the ultimate validation is always an in vivo model. They needed to see if this mechanism drives liver metastasis in a living organism.
So they used a splenic injection mouse model.
Yes. They injected a mixture of the engineered human endothelial cells and the colorectal cancer cells directly into the spleens of immunocompromised mice.
Why the spleen?
Because the splenic vein drains directly into the portal vein, which feeds straight into the liver. It perfectly mimics the physiological highway of human colorectal metastasis.
Okay, so when the cancer cells had their ITGB3 locks perfectly intact, what happened?
The mice rapidly developed massive metastatic tumors in their livers. The rogue blood vessel cells successfully facilitated the heist.
But here is the critical intervention. What happens when you break the receiver?
The researchers used RNA interference to silence the ITGB3 gene in the cancer cells, so the tumor cells could no longer produce the lock.
So the blood vessel cells are still corrupt. They're still pumping out massive amounts of the IGFBP5 key, trying to open the door.
Right. But the cancer cells were completely deaf to the signal. They had no lock, and the results in the mice were undeniable. The ability of the cancer to metastasize to the liver was severely crippled.
The liver nodules were significantly fewer and way smaller.
Exactly. Without that specific ITGB3 lock to receive the signal and trigger that relay race, the PI3KKT cascade, the tumor just couldn't execute the invasion, even with the blood vessels trying to help.
They completely neutralized the supply line.
Wow.
Okay, time for our final mini recap before we wrap this up.
Go for it.
Bullet point 1. The The signal jumps to the cancer cell via the ITGB3 lock, bullet point 2. This was proven with molecular docking, spatial transcriptomics, and the PLA assay. And bullet point 3, breaking the lock stops the PI3KKT engine and drastically reduces metastasis in living mice.
Perfect.
So my question to the listener is, if you were a scientist trying to cure this, would you try to destroy the key, the IGF-BP5? Would you try to plug the lock, the ITGB3?
Right.
Or would you try to cut the engine, the PI3KKT pathway?
It's a great question because that's exactly what oncologists are trying to figure out right now.
If you think about the trajectory we just mapped out, it is staggering. We went from a macro-level clinical crisis, patients dying because cancer spread to their liver, down to a single corrupt vanguard blood vessel cell.
And then down to a specific protein handshake, and finally down to a subcellular relay race that changes the cancer's physical shape.
It's amazing. And mapping that mechanism is exactly how we changed the paradigm of cancer treatment, right? Because for decades, the main strategy for metastatic cancer has been systemic chemotherapy.
Yeah, which is essentially carpet bombing the body with toxic agents to kill anything dividing rapidly. It causes massive collateral damage to the patient.
But understanding this specific key in lock presents a totally different therapeutic approach. It is precision sabotage.
Exactly. If you know that a tumor's ability to enter the bloodstream relies on this specific molecular handshake, you don't necessarily have to bomb the entire tumor. You can design a highly targeted antibody or a small molecule drug engineered to just cap the ITGB3 lock.
Or design a drug to neutralize the IGF-BP5 key before it ever reaches the cancer cell. You just sever the communication line.
Right. And if you stop the inside man from unlocking the door, the tumor stays localized. It remains a manageable, surgically removable mass rather than becoming a lethal systemic disease.
It really highlights a critical shift in how we view the tumor microenvironment. The cancer doesn't exist in a vacuum. It is deeply dependent on hijacking the healthy tissue around it.
Which leaves you with a really provocative question to consider.
Let's hear it.
We spend billions of dollars researching how to kill cancer cells directly. But if a primary tumor is this dependent on hijacking normal healthy cells, like these endothelial blood vessel cells, to survive and spread, what does the future of treatment look like?
Yeah.
What if the next generation of cancer therapeutics isn't focused on attacking the tumor at all, but rather on curing the healthy tissue of its corrupted programming?
Wow. So you restore the vascular barrier, fix the zipper, and simply let the tumor starve in isolation.
Exactly. It's a fascinating inversion of the problem. If the heist relies on an inside man, you don't necessarily need to shoot the mastermind. You just need to resecure your own personnel.
That is definitely something to mull over next time you hear about breakthroughs in cancer research. The most important discoveries aren't always about the tumor itself. Sometimes they're about the neighborhood it lives in.
Very true.
Well, thank you all for joining us for this deep dive into the microscopic mechanics of metastasis. We'll catch you on the next one.